Welcome to Unit Number 2, Part 3 of the MPharm Pharmaceutics video on molecular pharmaceutics, nanotechnology, and targeted drug delivery systems. This video is presented by Dr. Krishna Yadav. This video focuses on Unit 2, Targeting Methods, covering advanced targeted drug delivery systems from concept to clinical evaluation. We are in Section D, focusing on the evaluation of targeted drug delivery systems, specifically the physicochemical characterization. We begin with particle size and size distribution using dynamic light scattering or photon correlation spectroscopy. DLS measures hydrodynamic diameter based on Brownian motion and the Stokes-Einstein equation, although it is limited by its assumption of spherical particles. DLS measures the hydrodynamic diameter of nanoparticles in suspension. The process involves laser light interaction, Brownian motion analysis, scattered light fluctuation detection, and final particle size calculation. Next, we cover zeta potential measurement and morphological analysis. Zeta potential, measured by laser Doppler anemometry, determines the surface charge density, which indicates stability. Morphological techniques include TEM, SEM, and AFM. This illustration details zeta potential measurement, assessing colloidal stability, and morphological analysis techniques. TEM for internal structure, SEM for surface morphology, and AFM for 3D topography and mechanical properties, often in physiological fluid. Drug loading and entrapment efficiency are critical metrics. Entrapment efficiency measures process efficiency, while drug loading measures carrier capacity. High DL% is desirable as it reduces the required excipient amount. This diagram illustrates the calculation and process for both entrapment efficiency and drug loading. EE% is the drug in nanoparticles versus total drug added, and DL% is the drug in nanoparticles versus the total nanoparticle weight. In vitro drug release is studied using dialysis bags or flow-through cells in media simulating plasma or endosomal conditions. Kinetic modeling involves zero-order, Higuchi, and Korsmeyer-Pepys models to determine release mechanisms. The diagram displays the in vitro drug release method using a dialysis bag and the resulting data analyzed by various mathematical models to understand release kinetics. Surface and ligand characterization confirms conjugation. FTIR detects new bond vibrations. 1HNMR identifies specific proton shifts. XPS analyzes elemental composition of the surface. The various characterization techniques confirm ligand attachment. FTIR detects chemical bond vibrations. 1HNMR identifies chemical environments of protons, and XPS determines the elemental surface composition of nanoparticles. Ligand density is crucial for targeting avidity. Too high density risks immune clearance, while too low compromises binding. Stability studies follow ICH guidelines, monitoring aggregation, zeta potential drift, drug leakage, and chemical degradation. This visualization illustrates how ligand density affects avidity and the balance required for optimal targeting. It also details stability and shelf life studies, showing monitoring parameters like aggregation and chemical degradation. We now transition to Section 8, Biological Evaluation, starting with 8.1, In Vitro Evaluation, which assesses the nanocarrier's performance at the cellular level. In vitro evaluation includes cell viability, cytotoxicity, and cellular uptake studies. Cytotoxicity assays, such as MTT, measure metabolic activity. Cellular uptake uses flow cytometry and CLSM to quantify internalization and visualize localization. This visual summarizes the in vitro assays. Cell viability tests measure toxicity and therapeutic effects. Cellular uptake studies use fluorescent labeling and imaging to quantify and visualize nanoparticle internalization. Next, we examine receptor binding and competition assays. Competition assays confirm receptor-mediated targeting by blocking receptors with excess ligand. Transwell models simulate tissue barrier crossing, using cell monolayers like CACO2 for permeability studies. The diagram illustrates how competition assays confirm receptor-mediated targeting, showing high uptake versus low uptake in pre-treated cells. Transwell systems simulate biological barriers, measuring transport across cell monolayers. We move to 8.2, In Vivo Evaluation, starting with biodistribution studies. Animals are injected with the formulation, and drug content in major organs, including tumor tissue, is analyzed at time intervals. This maps the complete fate of the carrier. This image illustrates the biodistribution process and the resulting analysis, showing drug accumulation across various organs and tumors, confirming targeting selectivity in vivo. Next is pharmacokinetic (PK) and pharmacodynamic (PD) evaluation. PK studies measure plasma half-life and clearance. PD measures the therapeutic effect, such as tumor volume reduction, correlating drug exposure with biological response. The chart shows typical PK data, plotting plasma concentration over time, revealing changes in half-life, AUC, and clearance for different formulations. Targeting Efficiency Index, TE, quantifies drug accumulation selectivity at the target versus non-target sites. Relative Efficiency, RE, compares the performance of the targeted formulation against a non-targeted one at the same site. This visualization shows the calculations and interpretations for both the Targeting Efficiency Index and relative efficiency, providing quantitative metrics for targeted drug delivery systems. Imaging-based tracking techniques allow non-invasive monitoring in live animals. IVIS uses fluorescence. MRI tracks spion-loaded carriers. PET/SPECT uses radio-labeled particles for quantitative, real-time biodistribution. This slide visually summarizes the three main imaging techniques for in vivo tracking: IVIS, MRI, and PET/SPECT, highlighting how each method monitors nanoparticle localization. The final aspect is toxicological and safety assessment. This includes hematology to check blood compatibility, histopathology to examine organs for inflammation, and immunogenicity testing for antibody production against the carrier system. This concluding slide visually outlines the three key safety assessments: hematology, histopathology, and immunogenicity, confirming the safety profile of the targeted drug delivery system. This video utilized several textbooks and review articles. Textbooks covered foundational industrial pharmacy and pharmaceutical dosage forms. Review articles focused on polymer therapeutics, nanocarriers, and the impact of nanotechnology on drug delivery. Thank you for watching this video. Please like, share, and subscribe to my channel for more content.